Hello everyone and welcome. In this video we're checking out variant 1 of October-November 2020. Before starting, make sure to check out the free notes I have available for you in the comment section down below. Now let's go ahead and check out question number 1. Which row shows the correct order of sizes of these cell structures? Width of mitochondria and width of ribosome, width of cell surface membrane, width of chloroplast. Well, the chloroplast is definitely the biggest structure here. So that's 4. And then mitochondrion, and then ribosome, and then cell surface membrane. And remember the sizes. This guy over here has a width between 0.5 to 1.5. The chloroplast, I believe it's a lot larger. I think it's like 2 nanometers in width, and then or 2.5. And then the ribosome here is obviously it has an average, but like the 70S ribosomes are 18 nanometers, whereas the 80S ribosomes are approximately 22 nanometers, and so it's definitely bigger than cell surface membrane, which is only 7 nanometers in diameter or in width, and that's why it's 4, then 1, then 2, and 3. The diagram shows the stage micrometer scale viewed through an eyepiece graticle containing graticle, and small divisions of the stage micrometer scale are 0.1 millimeters, and the stage micrometer scale is replaced by the slide of a plant cell. And then what's the actual length of the nucleus of the plant cell? Okay, first things first, he gave us a calibrated stage micrometer unit. So we know the length of one stage micrometer unit. We don't have the length of the eyepiece graticle units. We want the length of the nucleus, and we have it against the eyepiece graticle. So we can use the eyepiece graticle to, basically find the number of units, find the number of eyepiece graticle units. And so now if we count this, it's going to be like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 units. So one nucleus is basically 10 eyepiece graticle units in length. So now we need to know how much are the 10 eyepiece graticle units, because these are not really right measurements. We need millimeter, micrometer, that kind of thing. We're not dealing with eyepiece graticle units. Okay, so what we're going to do is we know that 0.1, which is the length of this guy, is against 40 of the eyepiece graticle units, yes? So 0.1 is like 40 eyepiece graticle units. Obviously millimeters here. Now we're looking for 1 eyepiece graticle unit. How many millimeters is that? So what we're going to do is we're going to cross multiply. So you're going to have 40x equals 0.1, which means that 1 eyepiece graticle unit is actually 0.0025 millimeters, which obviously then you can convert to micrometers by multiplying by 1,000, and so you get x, which is 1 eyepiece graticle unit, to be 2.5 micrometers. So now if I have 1 eyepiece graticle unit and I know that each of these eyepiece graticle units is 2.5 and I have 10 of them, then I could just multiply 10 by 2.5 to get 25 micrometers for the length of the nucleus. 3. Insulin is a hormone synthesized from 2 polypeptide chains inside cells of the pancreas. Which row shows the cell structures that are used in producing insulin and releasing it from the cell? Well, to produce insulin, we need ribosomes, we need the endoplasmic reticulum, we need the Golgi apparatus, we need... do we need lysosomes? We obviously do not need lysosomes. Do we need the Golgi body? Yes, of course we do. The Golgi body is going to be there in post-translation modification, which is basically after you get the polypeptide chain being produced, you need to modify it so that it's now a functional polypeptide. So for example, for insulin, you need to remove the methionine, which is basically the first amino acid, okay, and then it's going to be ready. So yes, Golgi body. Lysosome, definitely not. The lysosome is a sac containing hydrolytic enzymes. It has nothing to do with releasing insulin. And obviously the endoplasmic reticulum, so that we can produce insulin. So that's why it's C. Now, the electron micrograph shows a cell structure in a eukaryotic cell. So which statements about the cell structure are correct? Okay, so we can see here that we have a centriole, and so he's saying here ATP is synthesized in the cell structure. No, okay, centrioles do not really synthesize ATP. The cell structure is made of protein molecules. Yes, it's definitely made of a whole lot of protein molecules. And the cell structure replicates during interphase of the cell cycle. Yes, well, remember, centrioles form a part of centrosomes. Centrosomes replicate during the cell cycle, and so centrioles also do, which is why it's 2 and 3. 5. 4 students were asked to match the function with the appearance of some cell structures in an animal cell, and the functions were listed by number. So if synthesis of polypeptides, synthesis of lipids, packaging hydrolytic enzymes that will remain in the cell. This is a very repeated question. We're going to go ahead and start with number 1. Synthesis of polypeptides is a ribosome. It's a non-membranous spherical structure, which is W. Synthesis of lipids is the smooth endoplasmic reticulum, which is a single-membraned organelle that's basically going to surround the inner closed cavity, which is V. Packaging of hydrolytic enzymes that will remain in the cell. Is it a double membrane interspersed with pores? Definitely not the nucleus. Is it a non-membrane bound cylindrical structure? Definitely not. Is it a membrane bound sacs arranged as flattened stacks? Yes, the Golgi apparatus. So that's Z. So it's W, V, and Z, which by the way is A. The DNA of typical prokaryotes is naked and circular. Which statements describe how the DNA of eukaryotes differ from the DNA of typical prokaryotes? Okay, the DNA in eukaryotes has a nuclear envelope around it and is a double helix. Actually, DNA in eukaryotes and prokaryotes is a double helix. The only difference is that in eukaryotes it's linear, in prokaryotes it's basically circular and, just like all over the place. And so only DNA of eukaryotes has a nuclear envelope around it and is circular. No, DNA of eukaryotes has proteins attached to it and is double helix. That's the general structure of DNA. It's the same all throughout organisms. Only DNA of eukaryotes has proteins attached to it and is linear. Exactly. Okay, you might think, okay, but don't prokaryotes also have proteins? No, but the prokaryotes have naked DNA, which means they don't have any histones in them, and so that's only D. A student carried out the Bendix test on different types of milk X and Y. A sample of each milk was heated to 100 degrees Celsius in a water bath with Bendix solution, and the time taken for the first appearance of color change was recorded, and the results are shown in the table below. So you could see table of milk and the time for the first appearance. You could see it's faster with solution X. He says here, which row shows the biological molecule the student detected in the sample of milk, and the sample of milk with the highest concentration of the biological molecule. Okay, the biological molecule present in the sample of milk, well, we don't really know what kind it is. It could be glucose, it could be fructose, it could be, most likely it's lactose because it's milk. Okay, but in the end it's simply a reducing sugar, and a sample of milk with the highest concentration of this biological molecule, it's definitely X, since the rate of at first color change is actually a whole lot faster, and that's why it's C. The diagram shows the structure of a monomer, and you can see it's down, down, up, down, so that's alpha glucose since it's a 6-membered ring. And then which molecules contain this monomer? So the molecules with this monomer are, the macromolecules of this monomer are glycogen and starch is actually made of 2 molecules which are known as amylopectin and amylose. So yes, amylopectin, glycogen, sucrose is actually made of both alpha glucose and fructose, so yes, which is why it's B. Which molecules are formed by the condensation which involves a carboxyl group? Okay, condensation which involves a carboxyl group. Now, carboxyl groups are found specifically in fatty acids and in proteins, or specifically in amino acids. And so, what's it called? Amylopectin, collagen, triglycerides. Obviously, not amylopectin. Amylopectin is it's going to be formed from a bunch of glycosidic bonds, which happen between hydroxyl groups, not between carboxyl groups. So remember, amylopectin here is a carbohydrate. It's definitely not going to be, it's either a triglyceride, collagen, or another protein. So collagen, beta globin, and triglyceride, exactly, because beta globin is another protein. Which row correctly matches the function with the structural features of cellulose, collagen, and glycogen or triglyceride? Molecule, function, fibrous. Okay, let's see. Firstly, molecule, cellulose, function is support, correct. Fibrous, yes, cellulose is fibrous. Actually, almost yes, all polysaccharides that we've dealt with are fibrous, and then when it comes to proteins, they are either fibrous or globular. Okay, molecules held together by hydrogen bonds. Yes, they are. Cellulose molecules are held together by hydrogen bonds. And then are they branched? No, they're linear, so that's why this part is correct. Okay, triglyceride, energy source. Yes, it is an energy source. Fibrous, it's not fibrous. Molecules are not held together by hydrogen bonds, and it's not branched, also true. So A is correct. Let's see the other options. Collagen, strengthening, fibrous, true. Molecules are held together by hydrogen bonds, no. Collagen is held together by covalent bonds. Branch chains, no. Cellulose, support, it's fibrous, it's not held together by hydrogen bonds. Okay, also wrong. So B is wrong. Collagen, glycogen, let's see. Collagen, strengthening, fibrous, molecules not held together by hydrogen bonds. C is wrong. Glycogen storage, not fibrous. Actually, glycogen is fibrous. Okay, molecules are held together by hydrogen bonds. Well, they could be, but it's not exactly polar, and it's different cellulose in structure. Branch chains, yes, branched. However, again, triglycerides are not held together by hydrogen bonds, so it's really A. Moving on to 11, the diagram shows the amino acids in a polypeptide, and he says here that you have C-terminal and N-terminal, and an enzyme catalyzes the hydrolysis of any peptide bond between a non-polar amino acid and an polar amino acid. Sure. So an nonpolar, which is basically this guy, and any polar, any of these guys. So any guys with like the circle here, this is going to be hydrolyzed. So how many small peptides and single bonds are what's it called formed by the action of this? Okay, so single amino acids and small peptides, how many? Let's see. Firstly, these guys are non-polar. These guys are this is polar, so this is gonna break as well as this. And so now we have these two chains. Now this is also going to break, and this is going to break. Here we're going to have a break, and then here we're going to have these two together. This guy is going to break here and there, and then same thing here, and then here, and then here. Okay, so let's count them. Firstly, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and then 12, no, not 12, 13, 12, and now 13. So we have 13 small peptides, and then when it comes to the single amino acids, we're going to have where's the first one? This one is a single amino acid, that's 1, and then we're going to also have 2 here, and then 3, and then 4, and so it's actually 4 and 13, which is D. Which properties of water reduce temperature changes inside cells? Okay, reducing temperature changes, okay, obviously cohesion, latent heat of vaporization, or specific heat of capacity. Okay, specific heat capacity does not really do anything to change. Latent heat of vaporization has to do with like the evaporation of water and how it's hard for water to evaporate and requires a lot of energy. So this has nothing to do with like cells. Okay, cohesion, I mean, water does like, connect together and everything, but again, temperature changes specifically relates to specific heat capacity, and so it's only 3. Which enzyme is extracellular? Amylase in saliva is an enzyme that catalyzes the breakdown of starch in the mouth. Amylase does break down maltose. It does not break down it also breaks down amylose, so yes, okay, it does break down starch in the mouth. ATP synthetase is an enzyme that's found in mitochondria that synthesizes ATP. So yes, ATP synthetase is an enzyme found in mitochondria. So he says here, which enzyme is extracellular? Amylase is extracellular. ATP synthetase is intracellular. DNA polymerase is intracellular. RNA polymerase is intracellular. It's really just amylase that's in the mouth. The graph shows how the concentration of the concentration of components of an enzyme catalyzed reaction changes with time. And so which line represents the enzyme with empty active sites? Okay, this is a very important question. He says that these are all the components of an enzyme catalyzed reaction, which means we need to eliminate all the graphs so that we're able to identify which one he is looking for, which is the enzymes with the empty active sites. The first one is pretty obvious. Concentration over time increasing so much and then reaching a plateau. It's definitely going to be the concentration of the product, whereas concentration of C decreasing so steeply is definitely going to be the concentration of substrates. And then when it comes to B, decreasing and increasing, well, let's see what D is. D is going to be increasing and then decreasing slightly more. So what do we think that D is? D is definitely the enzyme substrate complex, since it increases over time and then decreases. And then B is going to be the enzymes with free active sites. It keeps on decreasing in the reaction as the reaction goes, and then it starts to increase at the end of the reaction when the products reach a plateau, which is going to be, that's the answer, B. Which effects can non-competitive inhibitors have on enzyme-controlled reactions? Okay, non-competitive inhibitors, are they going to lower the Km value? Non-competitive inhibitors, they specifically work on lowering the Vmax. So remember, non-competitive inhibitors, they bind to the allosteric site of the enzyme, which is a different site, and they cause the enzyme to be non-functioning. So it cannot really bind to the substrate anymore, which means it cannot catalyze as many as many reactions, and so we never reach the maximum rate of reaction. However, the Km value still remains the same. Okay, reduce the concentration of the product. It's actually, yes, it's going to reduce the concentration of the product and reduce the rate of metabolic pathway, of course, so that's 2 and 3 only. Which processes can allow transport into or out of the cell? There's many processes. Active transport, facilitated diffusion, osmosis. Yes, of course, all of them allow the transport into or out of the cell. Plant cells were put into one of three different concentrations of sugar solutions: 10%, 5%, 2.5%, and the cells were left for 50 minutes and observed using a light microscope. Which statements are correct? Cell Y has a lower water potential than the sugar solution it was put into. Let's see. Cell Y actually has yes, it does have a lower water potential, and so that's why it's turgid, because it gained water. So what's it called? Since it has lower, and this here is higher, and it's probably 2.5%. Cell Z was put into a 10% sugar solution. Yes, it was placed into the most concentrated solution, so that here we have a lower water potential, here we have a higher water potential, and water leaves the cell, and that's why the cell is plasmolysed. So this is correct, and this is correct. Cell Z has a less negative water potential than the sugar solution it was put into. Actually, it has a less negative water potential. So having a less negative water potential means that you have a higher water potential. So yes, it is true, and that's why it's 1, 2, and 3. During telophase of mitosis, a scientist stains the chromosomes of a diploid animal cell with a fluorescent dye to observe the telomeres. The cell has 22 chromosomes. How many telomeres will the scientist observe for in one nuclei? Okay, in just one nuclei. Remember, there are 2 nuclei forming in telophase, and remember, in telophase, the chromosomes are going to be separated and placed in different nuclei, and the chromosomes are going to consist of just one chromatid. And one chromatid consists of 2 telomeres. So if we have 22 chromosomes, each with 1 chromatid and 2 telomeres, this means that we just multiply this by 2 to get the number of telomeres. And remember, 22 chromosomes, 22 chromatids, 44 telomeres. A methotrexate is a drug used to treat cancer. It can act as an enzyme inhibitor, preventing the synthesis of nucleotides containing thymine. Cells treated with methotrexate are not able to complete the cell cycle. A cell entering telophase is treated with methotrexate. Which stage of the cell cycle will be affected by the action of methotrexate? Now, since here it specifically affects synthesis of nucleotides, which means basically it stops nucleotides from being available, this obviously is going to stop DNA replication and protein synthesis in general. And so DNA replication happens specifically in the S phase, in the interphase. And so obviously here the part of the cell cycle that will be affected is going to be interphase. Which stage of mitosis is correctly described? In anaphase, chromosomes line up at the equator. No, actually this is metaphase. In metaphase, the chromosomes replicate. Centrosomes replicate during prophase, not in metaphase. And then the prophase chromatids move to opposite poles. That's in anaphase. And then in telophase, chromosomes uncoiled from chromatin. Yes. The diagram represents a nucleotide containing cytosine, and so he says here which statement about this nucleotide is correct. The carbohydrate would be ribose, not necessarily. Okay, it could be ribose or deoxyribose, since it did not specify the kind of nucleotide. The organic base contains nitrogen, of course, it's a nitrogen-containing base. Base pairing occurs with guanine, of course. Cytosine is a purine, that's wrong. Cytosine is Cytosine is a pyrimidine. Now, here with the answers, they don't really have 2 and 3 only, but which statements about this nucleotide are correct? The carbohydrate could be ribose. So yes, sure, it could be ribose. 1, 2, and 3, which is A. The section of DNA contains 73 base pairs. Number of bases on each strand: adenine, cytosine, guanine, thymine, strand 1, strand 2, and everything. And he's saying which row represents the correct number of bases. So V. Okay, now firstly, 20, okay, so he's looking for V and Y and Z. Now, he's gave you strand 1 of adenine, strand 2 of, adenine. And we know that on strand 2 of, adenine, if there's 21 on strand 2 of adenine, there should be 21 on strand 1 of thymine, because adenine is complementary to thymine. And if there's 29 on strand 1 of adenine, then there should be 29 on strand 2 of thymine. And then when it comes to cytosine, here, since we have 14 on strand 1, this should also be 14. And then to calculate the rest, he said here they have 73 base pairs. So 73 base pairs, you have already a whole bunch of them. All that's left is cytosine and guanine. So remember, a base pair is two bases paired together, right? So it's two bases. So 73 times 2 is 146. 146 minus, 29 plus, obviously 21 plus 14 plus 14 plus 29, 21 plus 29. And then if we subtract all this, we're going to be able to get the rest of cytosine and guanine, and so this gives me 18. So 18 is left, but don't forget 18 is cytosine and guanine. We need each one of them, so we divide by 2, so there's 9 cytosine on strand 1 and there's 9 guanine on strand 2 right here. And so V is equal to 9, X is equal to 14, W is equal to 21, and Y is equal to 9, and then lastly Z is 29. That is A. Now the photograph shows a section of a stem which labeled part is the xylem. Okay, xylem is this section, as you can see over here. He's labeled a whole bunch of things, but yes, okay, we could see clearly the xylem vessel elements here. So moving on, some fungi cause wilting in crop plants by growing within xylem vessel elements. Which process would be directly affected by these fungi? Okay, so he's saying here that they grow within the xylem vessel elements, which obviously could affect transport of water, transpiration in general. So cohesion between water molecules is going to be affected, while the fungi is going to be within the xylem. So water is going to, I think adhesion is going to be the one affected. Let's see. Development of root pressure is going to be just fine because it's in the root, and he said here it's just within the xylem vessel elements. Mass flow during movement of assimilates. There's no assimilates moving in xylem. Uptake of water by root hair cells. Again, it's something to do with the root, not with the xylem. So it's definitely the cohesion between water molecules. It could basically alter how the water molecules are going to bond, since there is not much space left for them to bond together. Which features have a role in the transport of water in xylem vessel elements? Capillary action, adhesion, hydrogen bonding. Actually, all of those. The loading of sucrose into companion cells involves a number of processes. Which process is active? The movement of hydrogen ions into the cytoplasm of companion cells. Okay, so he's saying here that the loading of sucrose into companion cells involves a number of processes. Yes, so the movement of hydrogen ions into the cytoplasm of companion cells. Actually, they move out of the cytoplasm of companion cells and then back in. But like the process that's active is going to be moving out, okay, because they're pumped out and then they move back in by facilitated fusion, which is obviously passive. So the movement of hydrogen ions out of the cytoplasm, yes. The movement of sucrose into the companion cells is by co-transport, which is actually indirect active transport, which we know is also passive. And the movement of sucrose out of the companion cells, sucrose does not leave the companion cells, and that's why it's B. The statements describe which events that occur during the movement of sucrose. Let's see. Which sequence correctly describes what happens in the plant at source during the movement of sucrose? Okay, so at a source, what happens first is sucrose. Okay, let's see the first option. Hydrostatic pressure is decreased, hydrostatic pressure is increased. Sucrose is loaded into the phloem vessel. Sucrose is unloaded from the phloem vessel, and then water moves by osmosis. Now he's saying here specifically at the source, so I know that some of these are actually wrong and are not going to be included in the answer to begin with. Water moves by osmosis into the phloem. Water moves by osmosis out of the phloem. Water potential decreases in the phloem. Water potential increases in the phloem. So let's remember what happens at the source. So this over here, I'm going to draw, Yes, okay, so this over here is the sieve tube element, and this is the companion cell. Okay, now we can see here that the companion cell is supposed to have plasmodesmata and everything, so these openings. And then besides that, the companion cell over here is going to allow sucrose to enter and then move into the phloem. Now once sucrose is here, this is going to be at the source. Okay, sucrose is going to be here. This is going to decrease the water potential. So once water potential decreases, water from the xylem is going to enter right over here through the pith, and then once water is going to enter, this increases the hydrostatic pressure, and then this allows the material or assimilate to move down or up, of course, according to the place by mass flow. Okay, so hydrostatic pressure should be increased. Sucrose is loaded into the phloem vessel, and water moves by osmosis into the phloem, and water potential decreases in the phloem. So the first thing is 3, then water potential decreases, then water moves into it by osmosis, and then hydrostatic pressure increases. 3, 7, 5, 2, which is A. Which are present in the walls of capillaries? Endothelium, smooth muscle, elastic tissue, or collagen fibers. Actually, the only thing available is endothelium cells. Okay, they don't have smooth muscle or elastic tissue or collagen. They're just a layer of single cells. Okay, now the photomicrograph shows 3 white blood cells labeled X, Y, and Z. So he's saying which row identifies these cells correctly. So Y is the neutrophil, Z is the monocyte, Z is the lymphocyte, and then X is the monocyte. Okay, let's see. Cell X is the monocyte, cell Y is the neutrophil, neutrophil, and cell Z is a lymphocyte. And you're gonna say, how do I identify them? First, it is a very repeated question. You should identify them based on what you've memorized from the book. But in general, Y, having a lobe nucleus, being a granular, is going to be traits of a neutrophil or a phagocyte. Z, having a large nucleus that fills out the cell, is a trait of a lymphocyte, unless you are a neutrophil that does have a large nucleus. However, it's very, very huge, and still it's not like filling it out to the brim. So since it's not filling it out and it's very huge, this is definitely either a monocyte or a macrophage. But since we are within the blood still, this is going to be a monocyte. Okay, once the monocyte leaves and the blood and goes into the tissue fluid, it's going to be called a macrophage. Which sequence of letters correctly identifies the order of events during the cardiac cycle? Atrial walls contract. Okay. So the first thing is going to be the sinoatrial node. The sinoatrial node is going to spread the wave of excitation, which is going to cause the atrial walls to contract, which is T, and it's going to reach the atrioventricular node, which is going to delay the impulse by a fraction of a second, which is U, and then it's going to be passed. Now, the reason that the atrioventricular node delays the impulse is because we don't want the atria and ventricles to contract at the same time, and then a wave of excitation passes down pericardial tissue. After that, the ventricles will contract. So it's X then T, then V, then U, then W, then Y. Let's see, W, then Y. Okay, yeah, that's exactly what I wrote, right? X then T, then V, then U, then W, then Y. Yes. Okay, what about this guy? X then T, then V, then W. That's wrong. Or X then T, then U. That's also wrong. Or X then V. That's also wrong. And that's why it's A. The graph shows the effect of 3 different partial pressures of carbon dioxide on the oxygen dissociation curve for the human hemoglobin. And so you can see that we have different partial pressures of CO2, and as you increase the partial pressure of CO2, the graph is shifted to the right, okay, and the affinity of hemoglobin to oxygen is going to decrease. Okay, so what effect does decreasing the partial pressure of carbon dioxide have on hemoglobin? Decreasing the partial pressure of carbon dioxide, so like in graph X with like the lowest partial pressure, it actually increases the affinity of hemoglobin to oxygen, therefore increases the percentage saturation of hemoglobin with oxygen. So it is less efficient at taking up oxygen, less efficient at releasing oxygen. Actually, no, it's more efficient at taking it up, less efficient at releasing it. Okay, that is C. Now, what is correct about the transport of carbon dioxide by the blood? The enzyme carbonic anhydrase catalyzes the formation of carbonic acid in the red blood cells. This is true. Carbon dioxide diffuses from active cells into the red blood cells and reacts with water. Carbon dioxide actually diffuses, yes, from active cells and then to red blood cells, and then it reacts with water to form carbonic acid, which is catalyzed by carbonic anhydrase. Carbonic acid dissociates, forming hydrogen ions that combine with hemoglobin to form carbamine hemoglobin. No, it forms hemoglobinic acid, which is why it's 1 and 2 only. Which statements about all bronchioles are correct? They have goblet cells, they have seated cells, they have muscle tissue. Okay, they don't have goblet cells. Some of them do have seated cells and like in very low amounts. And then lastly, yes, they do have muscle tissue, which is why it's 2 and 3 only. The graph shows the volume of air breathed out quickly with force, followed by a deep breath in 3 different people, X, Y, and Z. And then you can see here what is a possible explanation for the difference in volume of air breathed out by these people shown. You have the volume of air breathed out by these people. Okay, so the difference, okay, so here we have chronic bronchitis or emphysema or normal lung function. This is long out of your syllabus. Emphysema is not part of your syllabus anymore, which is why 34 is eliminated. And then 35, the diagram shows some of the pathogens that cause disease in humans and some ways that they are transmitted. Okay, so you can see pathogen, bacterium, protoctist, and morbillivirus. So method of transmission. He's saying here what is correct pathogen and method of transmission for tuberculosis. Tuberculosis is caused by a pathogen which is a bacterium, either Mycobacterium bovis or Mycobacterium tuberculosis. Method of transmission is via airborne droplets, which basically are coughs and sneezes. So 1 and X, which is B. The proportion of local population who have malaria in area R is higher than the proportion in area S. What factor caused the difference? So the difference in the proportion of local population. Okay, so the difference in area R to area S could be that it's more humid, could be that it's hotter, could be that area itself, I guess like in general maybe it's a bit more rainy, that kind of thing. So let's see. Area R has a more humid climate than area S. Totally. Area R is near the equator than area S. Well, with climate change you can't really tell that like just because some country is near the equator that means it's a humid country. We can't really say this. There's a higher population in area R than S. This would be right with things that are airborne, for example, like let's say tuberculosis, but not with what's it called, area R has, like in the case of malaria, because malaria spreads primarily with mosquitoes, which are the vectors in this situation, and so the more mosquitoes, the more it spreads. It's not about the people itself. There is less sewage treatment in area R than area S. Less sewage treatment would be okay for waterborne, foodborne infections like cholera. Okay, because then, for example, sewage treatment is not good, water is contaminated, and so this disease is going to spread. But again, malaria spreads by a mosquito vector or by blood transfusion, so that's A. Species X is a single-celled eukaryote. Species X has been genetically modified to produce penicillin, which does not harm the cell walls of species X. So what do we conclude from this information? Obviously, they are eukaryotes. They do not have cell walls made of peptidoglycan, so that's why they cannot be harmed. So the cell walls of species X are chemically different to those of bacteria, of course, because if they were the same, then we're definitely going to be affected by penicillin. The cell walls of species X are made of peptidoglycan. No. Again, because bacteria cell walls are made of peptidoglycan. The cell walls of species X are made of cellulose. It can't really be concluded from the information. It could be, but not necessarily. It could be made of chitin, it could be made for any other material, so it's 1. Okay. When exposed to an antigen for the second time, memory cells stimulate a secondary immune response, which correctly shows the secondary immune response. Okay, the memory cell is going to, obviously after clonal selection, are going to undergo clonal expansion, where they're going to divide and then they get specialized into turning into plasma cells, which produce antibodies. Okay, so it's really similar to to the primary immune response, except it's faster, because clonal selection does not take that much time anymore, and does not take as much time for the cells to produce antibodies in general. And so the memory cell is going to turn into antibodies. No, is it going to turn into plasma cells? Well, okay, let's divide into plasma cells and memory cells. Exactly. It's going to divide into memory cells divide into plasma cells. That's C, or secrete antibodies and divide into memory cells. Remember, antibodies are secreted by plasma cells, so there are no antibodies without plasma cells. Monoclonal antibodies are produced for use in diagnosis or treatment of disease. To obtain the antibodies for an antigen, a mouse is injected with an antigen. And so some of the events in the production of monoclonal antibodies are listed. So he first injected the mouse with an antigen. After that, the mouse is going to carry out some immune response, and it's going to produce some plasma cells. Now, these plasma cells are going to be taken from the mouse by taking its spleen. So first, we're going to take the mouse's spleen, so that's 5. After we take the mouse's spleen, we're going to get the plasma cells, and we're going to fuse them with cancer cells, which are melanoma cells, to produce hybridoma cells. So plasma cells are fused with a cancer cell to form a hybridoma. Yes. After that, once we have the hybridoma, we're going to actually get the hybridomas to produce all the antibodies that we want. After we produce the antibodies, we're going to identify which ones are the ones necessary. Now, he says here that actually B lymphocytes that recognize the antigen multiply and become plasma cells. So yes, actually 3 comes before 5, since B lymphocytes produce the plasma cells, then the plasma cells are removed. Now, we've got the hybridomas. The hybridomas divide and secrete antibodies, and then the secreted antibodies that we require are going to be identified and cloned, and that's going to be 2, 3, 5, 1, 4, 2, which it doesn't go to the end, but it's just 3, 5, 1, 4. That's B. Which row describes passive immunity triggered by an antigen? It's not triggered by an antigen. It's just that there isn't any B lymphocytes being triggered by an antigen. It's just antibodies being injected into the patient, and so that's wrong. Involves an immune response. No, there isn't going to be any clonal selection or clonal expansion. There will not be a primary or secondary immune response. Memory cells we produced. There's no memory cells and passive immunity. There's definitely no permanent protection. So that's D. Okay, now this is the end of today's video. I hope this video was useful to you guys. If it was, make sure to like, share, and subscribe. And I'll see you guys in my next one.